Hello, everybody. Welcome to 300 Years and a Million Dead Plants, a behind-the-scenes virtual tour of the University of Cambridge Herbarium here in Cambridge. My name is Sabrina Gonzalez-Jorge. I am a plant biology at the Department of Plant Sciences, and I absolutely love the treasure trove that the University of Cambridge Herbarium is. Not only does the herbarium contain an amazing collection of specimens that are the holy grails of plant science and history, but it also contains a priceless or is a priceless resource for modern day researchers as well as plant aficionados, society as a whole, and humanity really. So today you will have a chance to see a little bit behind the scenes at the herbarium. You will meet its fantastic curator, Dr. Lauren Gardiner, who will basically focus today on showing you some of the historical collections that we have, and then we will have some live Q&A questions from you. So please do come up with as many questions as you like. And there's a Q&A session, so Lauren will try to expand on the relevance of the herbarium as a resource and the use of the specimens that we have in modern day research. But before we get on with the tour, I just wanted to say there's a few little housekeeping details that we must run through. This event has a Q&A box because we're in webinar mode, which means that any questions that you have, please put them in, and we will try to go as many as we can towards the end of the tour. There's also a chat function, so by all means, you can communicate with each other during the event, and please feel free to use this, but be mindful of any inappropriate use of the chat because it may actually result in the function being switched off and participants being ejected from the session. Lastly, in the unlikely event of a fire, go to your own nearest emergency exit. Just do it, leave it, it's fine. We'll be here when you come back. And so without further ado, I would like to introduce you to Dr. Lauren Gardiner, who is the curator of the University of Cambridge Herbarium.
Hello and welcome to the Cambridge University Herbarium. My name is Lauren Gardiner and I'm the curator of the herbarium here. The herbarium is part of the Department of Plant Sciences and it is a collection of an estimated 1.1 million herbarium specimens collected over more than 300 years. The collection has developed over more than 300 years from the collections of different botanists who have worked here at Cambridge or been associated with Cambridge in some way. Alumni have left us their collections. We've had various collections which have been purchased over the years and added to the herbarium. But in recent decades, the herbarium has not really been used very much and it's sort of fallen off the map a little bit. One of the things that we are working to do now is to make the collection much more active and accessible for researchers, for students, for members of the public. So over the next few years, we're going to really work to make the collection much more accessible and viewable by members of the public and researchers. The site where the herbarium is based is within the Botanic Gardens and it's within the Sainsbury Laboratory. It's not really a site that can be open for people to come in and out of. We don't really have an exhibition space, but by doing tours, by doing online activities and making our specimens available online, we should be able to make the collection much more open. So the herbarium was founded by John Martin, who was the second professor of botany in Cambridge in the 18th century, and he was succeeded by his son, Thomas Martin. Between the two of them, they accumulated a large herbarium, Hortus siccus, of dried pressed plants. And I'm going to show you some of those specimens. It was really John Stevens Henslow, though, who became the fourth professor of botany in Cambridge in 1825, who made the collection a much more scientific, modern teaching and research collection. John Stevens Henslow was also really the founder of the modern botanic garden here in Cambridge. And in a similar way, he laid gardens and the herbarium out systematically according to the new theories of taxonomy and classification. And he really used specimens and living plants in his research. Henslow is really known today as being Charles Darwin's mentor. He was his tutor when Charles Darwin was a student here in Cambridge. And it was Henslow who got Darwin really interested in diversity and observing the variation in nature. It was also down to Henslow that Charles Darwin ended up on the voyage of the Beagle. And we have Charles Darwin's botanical specimens here in the herbarium because Darwin sent those specimens back to his tutor, Henslow. Henslow distributed some of the specimens to places like Kew, but the main collection stayed here in Cambridge, and we have those specimens today. So now I'm going to take you into the herbarium. I'm going to give you a little bit of an overview of the space and how the collection is arranged and the facilities we have here. And I'm going to show you some specimens and show you our new digitisation studio. So before we actually go into the workrooms of the herbarium and the collection space itself, we have a prep room. The prep room is where we can prepare specimens which have been made either in the field, or they've been sent to us, or they've been made in the garden. The specimens are dried and pressed, and then they are all frozen in this big chest freezer. The specimens are frozen for at least 3 days at -30 degrees, -40 degrees. And that will kill any pests which are on the specimens. This is our best defense against specimens getting eaten by various pests that will destroy the material ultimately. In the past, we were able to use a lot of different chemicals to kill pests and to keep pests away. Obviously now we can't use many of those chemicals. They're not particularly safe for humans. And this is our most usual line of defense against pests. If we make specimens well, if we dry them, press them, we keep them dry and we keep the pests away, keep the temperature fairly stable and the humidity stable, specimens can last for hundreds of years, particularly if they are not exposed to light. So you'll see when we look at the specimens, they are kept in folders, they are kept closed, and we open them up to study them, but we don't leave them out on display for long periods of time. So this is the herbarium collection itself. So this room, we are able to keep the temperature and the humidity between a range that is good for storing specimens long term. Most of the collection is in compactor units, and I'll take you down the centre of the room so you can see some of those, but it's movable roller stacking, which means we can have a lot of specimens within a smaller space than you would otherwise be able to. The specimens are laid out in geographic areas, and also we have quite a few separate collections associated with particular individuals. So the Darwin specimens are kept separately, for instance, and Oliver Rackham's herbarium is kept separate for various reasons. Around the outside of this part of the room, we have these drawers, and these drawers are filled with packets. So the packets inside the drawers contain small specimens, and then on the outside we have the collection information for each specimen. This is obviously a much more compact way of storing smaller specimens. This is particularly used for bryophytes, so mosses, liverworts, hornworts, and also we have some lichens and we have some microfungi and other taxa in these collections. One of the most important sets of specimens we have here is our type collection. Now, type specimens are the specimens that names are attached to. So when a plant is described, a specimen is designated as being the type specimen for that plant name. We know that the herbarium here in Cambridge is particularly rich in types. We have an estimated 50,000 type specimens out of our 1.1 million herbarium specimens. Some of them have been identified in the collection over the years by researchers, and they've been pulled out and they've been put into these red folders. So here we have the red folders kept separately. And these are all type specimens. So these are some of the most important ones that many researchers inquire about, want to see when they visit the collection. These are our most important specimens, really. However, this is the tip of the iceberg. This is not 50,000 specimens here. Most of the specimens, the type specimens, are within the main collection, and they just haven't actually been identified as being types and designated. So when a researcher comes, studies a particular area of the main collection, they very, very commonly pull out newly designated type specimens, or specimens which have not previously been recognised as types and put into these red folders. So now we'll walk down the centre of the main hall.
I mean, that's the wrong spelling of centre. We're in Britain.
So here on one side, most of this side up until this point is the collection, which is Great Britain and Ireland. And this herbarium is extremely comprehensive. It's one of the best collections for Great Britain and Ireland herbarium specimens there is. And it was laid out by Peter Sell and Gina Morell when they wrote their 5-volume Flora of Great Britain and Ireland. This is a really comprehensive, rich collection of specimens. Down from this point to the end is the Flora Europaea herbarium. And this was one of the herbaria which was used in the writing of the multi-volume Flora Europaea. That collection hasn't really received very much attention for quite some time, and contains some very interesting things for sure. On the other side, all of the compactors along here are in the World Herbarium. Now, this collection, again, has not received very much attention or study in recent decades, but this is the part of the collection that is very, very rich, particularly in undesignated type specimens and specimens from 19th century and early 20th century collectors. This is a really interesting collection for me as a tropical botanist, a particular interest in tropical botany. And this is the area of the collection where, if you want to find type specimens, researchers definitely want to start looking. So as I said, some of the collection is laid out in separate individuals' collections. And we have some of the collection of John Lindley separate here in, there's 2.5 sides of compactors towards the end of the World collection. The rest of the Lindley collection is actually integrated into the main World collection. But within there, we have the majority of John Lindley's herbarium specimens. Now, John Lindley was the secretary of what became the Royal Horticultural Society. He described thousands of new plants to Western science, and particularly plants of horticultural interest. His specimens often have botanical artwork on them. There are many, many type specimens in there, and this is a really rich and exciting area of the herbarium. So as with many herbaria, we do have a sizable collection of material and specimens which have never really been researched at all. They are sometimes still in the original newspapers. They often don't have the labels of the information attached to them, but hopefully the information is with them, or we can find it from other archive sources. But this is material which needs to be curated and integrated into the main herbarium. Now, there are many boxes in this area, and many of them have names of very interesting, very important botanists, parts of the world, periods of time. So again, this is a very interesting area for researchers in particular to come, historians to come and look and see what specimens we have. So this is the herbarium's new digitization studio. This is where we have a couple of Macintosh computers and cameras and light boxes, and also a scanner set up. This equipment means that we can image our specimens, high resolution, very standardized lighting, scale bars, color charts, and we can make these images of the specimens which we can then either deliver to researchers on request, but increasingly and ideally, we get the specimens available for anybody to access online. And so over the next couple of years, we will be really making this happen for the Cambridge University Herbarium, starting with particular discrete parts of the herbarium with particular research and stories to tell based around the specimens. So we will be imaging our specimens and we will be making them available for anybody to access, and they will be on the Cambridge Digital Library platform. So watch this space because there's going to be a lot of progress in this area over the next year for sure. So I'm going to show you some specimens now and sort of tell some of the history of the collection as well as I go through. So inside the folders of specimens, we have our sheets inside extra protective folders. This is one of our specimens from the Martin Herbarium. So this is an anemone, which would have been collected in the early 18th century. Here you can see we've got the herbarium specimen itself. So this is the original plant material, about nearly 300 years old, this plant. We've got the original labels, the collection labels, and then later on, other labels have been added with more information. So we have the annotation of Dr. Fuchs, who's the individual who collected this specimen and sent it to John Martin. And then we've got a polynomial name, so a Latin description almost, which was used to refer to this particular plant. That's written by John Martin. And then we have Thomas Martin adding the Latin binomial. So this is the system that Linnaeus popularised of having a genus name and a species epithet, a two-part Latin name. Now, John Martin assembled much of his collection before Linnaeus popularised that system, and that system started being used consistently. And it was Thomas Martin, we think, who added a lot of those Latin Linnaean binomials later onto the specimens. This specimen was one of the ones that Henslow, when he became professor of botany, rescued. Basically, when Henslow became the professor of botany in 1825, he inherited the Martin Herbarium, and he found that a lot of it was in bad condition, a lot of it was pest damaged, water damaged, and a lot of the material had to be thrown away completely. He salvaged what he could and he remounted many of the specimens. And so this is a specimen where we've got a 300-year-old plant, but the paper it's mounted on is Henslow's paper, which is about 200 years old. And he added the labels, he transferred the labels across where he could too. This writing down in the bottom corner is Henslow's handwriting, and that's where he's gone through and he's added the up-to-date names himself as well. And above you can see a little tick and a date and some initials, and that is Peter Sell's handwriting. And Peter Sell worked here for many, many decades in the 20th century through to the early 21st century. And he annotated specimens too to update the names, to verify the identifications too. So this kind of specimen tells a lot of history, and specimens do change over time. People add new information to the sheets themselves. They're not preserved in aspic like one might think of for such historic material. But yeah, they change and new information is added over time. Some of the specimens from this period were collected because they have particular economic uses or medicinal uses. And this is one where we have a whole page of notes on the specimen about how to identify the plant, how to prepare the plant, and how to use it medicinally. We have the plant material, and then we have the label attached, the original label with the common name, and then a later name which has been added, a Latin name. And many specimens from this time period would have been collected with particular uses in mind. Here's another specimen with economic and cultural information on the specimen or connected with the specimen. This is one of the specimens in the Haddon collection. Now, the Haddon expedition of 1898 to the Torres Straits Islands collected a huge amount of cultural material that's in the Museum of Archaeology and Anthropology and in other collections, not in Cambridge. But the plant specimens which went with those more anthropological collections are here in the herbarium. And these specimens are another example. One of the collections is a discrete collection of a particular expedition, which we will image and make available for researchers so that these specimens can be connected with all the other materials which are out there, and the whole expedition's resources can be compared and brought together. So Henslow really enriched the herbarium when he was a professor of botany. He took the Martin collection and he salvaged what he could. He updated the names, he annotated the specimens. He also added a huge number of specimens to the collection himself. So here's one of Henslow's sheets, and Henslow has very distinctive specimens in many cases. And this is a nice example. This specimen shows multiple individuals of the same plant collected and assembled in what he termed a collated sheet. So here we have a population of one particular plant with all the different size variation. We also have some others which are growing with slightly different growth habit. And he very, very neatly annotates his specimens. Henslow's writing is really legible compared to many botanists of this sort of time period, of the early 19th century to mid-19th century. Here you can see he's got the plant name, he's got the location. This is Gamlingay Heath, which people based in Cambridgeshire will know, hopefully. And we've got the date of the collections of the two sets of plants and who collected them. Again, you can see a label from Peter Sell, later on updating the names. So Henslow's specimens also includes some really beautiful artwork. Some of it's his own artwork, some of it's artwork from other sources. This specimen is particularly lovely. This is Esculcia californica, the Californian poppy, which we have growing in the Botanic Garden here, and many horticulturalists will know from their own gardens. Here we have an illustration that came from the publication of this species. And if I open this volume from 1842, you can see here is the plate. This is a hand-coloured plate in this volume. And here is the description of the plant. Henslow has taken a copy of this plate, again hand-coloured, it's hand-painted, and he has annotated it. And you can see on the illustration, he's added labels of the different parts. We also have plant material, and this is plant material that he grew in the Botanic Garden. And he's got the normal form, as he says, that the usual state with the mitriform calyx, which is here, which you can also see mitriform calyx in the illustration. But here he has the monstrous calyx, as he calls it. This is where the calyx is splitting and growing very, very differently. And this was something that Henslow was doing a lot. He was observing nature, he was observing variations and distributions and mutation, what we now know are mutations, but what he called monstrosities. Also hybrids, he was very interested in hybridization. Henslow also produced lots of teaching sheets. His teaching methods were really innovative at the time, and he used illustrations a lot in his teaching, in his lectures, and in his demonstrations with students. Here's one of his teaching sheets, and we have many of these which have not been made available to date, but will be. But here we've got illustrations which have come from different journals, books sent by correspondence from his own library. He's assembled them in these sheets for each different plant or each different plant group, and he's annotated them with all the parts. And there are records of his teaching where it's written about that Henslow would pass around these demonstration sheets showing all the different forms and stages of life for a particular plant. We also have some of his original artwork. So here we have some watercolours of fungi, which were completed by Henslow. These are all fungi which Henslow observed in the Cambridgeshire area. We've got Bottisham Common, we've got Gogmagogs, different areas. Again, people from the Cambridgeshire area who are familiar with this area will know these locations. And these are from the mid 1820s, this is 1825, 1826, you can see. So another really exciting part of the herbarium, which I mentioned right at the beginning, is the Darwin collection. The Darwin collection is around 1,000 sheets, 1,000 specimens with plants attached to them. Many, many more plants than 1,000 sheets, because again, they're often collated sheets like this one. So here is a specimen, several specimens collected by Charles Darwin on the voyage of the Beagle. These were collected in Patagonia, these grasses. And these are collections of a grass, of what Darwin thought were the same species. Actually, now they're considered to be two separate species, growing in the same place at the same time. And you can see how, in the way that Henslow collected and presented his specimens, we've got multiple instances of that same plant, or what Darwin thought was the same plant. Now it's Henslow who actually put these together on the one sheet as a collated sheet, and this is Henslow's handwriting on the sheet, not Darwin's. But these are the specimens that Darwin collected, and he's collected in the same way that Henslow collected. There's no real reason why he would have collected multiple specimens of the same plant from the same place at the same time, unless he was looking out for these kinds of differences. Here's another Darwin specimen, and this is a specimen from the Galapagos Islands. Here we have a fern, and this is a fern species that Darwin collected in October 1835. And you can see he's folded some of the leaflets over so you can see both sides. And there's a letter in the Cambridge University Library where Henslow writes to Darwin, and he tells him to, if a specimen has got different features on different sides, to fold over the tips, particularly if things are too big to fit on a single sheet. And there's a little diagram where he shows this, which is rather interesting. Again, Darwin was looking out for differences, unusual things, and so from the same clump of this fern fronds, he also collected another sheet that is from the same plant, but you can see here it's growing in a very, very different way. And this is what Henslow would have described as a monstrous form. This is a mutation. So this is one where instead of having these pinnate leaves shaped like a feather, we've got a palmate leaf with all of the leaflets coming from the same point. Again, we've got the folded over tips, which is quite nice. You can see the upper and lower surface. Now, we do find specimens in the collection, even of people like Darwin, that we didn't know we necessarily had. And this is a slightly different type of specimen. This is a fungus specimen, not a plant. And it was originally collected and preserved in alcohol. So this is why it's in a jar. Now, this specimen was at the back of a cupboard, and the seal had long ago split, and all of the alcohol had evaporated off. It was pretty filthy, but we had a conservator look at the specimen and clean it up and carefully preserve it. And they were able to basically clean it up, make it look like it looks now, which is much, much clearer, including the label. And we realised that this is actually one of Darwin's specimens, again from the voyage of the Beagle. And this is also part of the type material for this particular fungus. So this is a fungus, Sitaria darwinii, named for Darwin, which Darwin collected in Tierra del Fuego. And most of the the rest of the specimen is at Kew, and is designated as the type specimen. But actually, this is also part of that material. So this is also type material for this species. This was just found a couple of years ago here in the collection. We also have collections from some other really significant botanists in this collection. This is a specimen collected by Alfred Russel Wallace in Borneo. We have the largest botanical collection of Wallace material that survives. It's a collection of 41 fern specimens that he collected. He seems to have collected them from a particular point, a particular location, population diversity survey of all the fern species growing in that area at that one time. There's a whole story behind these specimens, but again, they were only discovered relatively recently, within the last decade. So you might have noticed that the specimen we just looked at, the Wallace specimen, is in one of those red folders, and that is because it's one of the type specimens. So that is the specimen that that species was named and described in the scientific literature from. This is another of the type specimens, and this is from John Lindley's herbarium. So I mentioned how Lindley particularly compiled specimens of horticultural and ornamental and economic interest. This is one of Lindley's type specimens, and this is a succulent, Echeveria acutifolia. If I put it on a sheet and show it to you a little bit more closely, you can see here we have the plant material and we have the original label attached to it, which says it was collected by Hartwig in Oaxaca in Mexico in 1841. And then we've got a hand-coloured illustration again here on the sheet. If we go to the original literature, so this is the volume in 1842, published by John Lindley, we can see the original description of this species, and you can see that same plate attached. And this description talks about Mr. Hartwig collecting this in Oaxaca, Mexico, in the 1840s, sending the plant material back to England. So that specimen was in a red folder. That has been identified as being a type specimen previously. So it's been pulled out, it's been put into that, curated into that red edged folder. This is a specimen which had not previously been identified as being a type specimen. This was found in the collection by some students a couple of years ago. They started going through the Lindley collection and pulling out all the specimens where there was artwork on the specimen, on the basis that if someone had bothered to add an illustration, it slightly increased the chance that that was possibly a type specimen or of particular interest. This was one of the ones that they found, and I think this is particularly beautiful. This is a relative of hydrangea. It's now known as Dicroa, but it was named Adamia versicolor when it was first described. We've got this beautiful piece of artwork. It's not finished, it's half completed. You can see the line drawing, you can see the painting itself. And there's very, very little information on this specimen. It says HHS, September 1846. HHS is Herbarium of the Horticultural Society. That's what became the Royal Horticultural Society. There's also a name. Again, this is John Lindley's handwriting. If we look up the original publication information for Adamia versicolor, we find the original publication, and it talks about a plant being collected in Hong Kong by Robert Fortune. It describes this plant, it describes this inflorescence, and there's a plate, which is the finished plate of this illustration. It describes how this plant was collected and it was sent back to the Horticultural Society of London, and it flowered in the collection of the Horticultural Society in September 1846. So this is the type specimen of that species. And you can see how rich this collection is. This is a rhododendron from Borneo. This is very beautiful specimen on multiple sheets. You can see the flowers, the leaves, and then there's several original illustrations attached to these as well. We have the red form and we've got the yellow form as well. And this is the part of the type material for this species. And just to show you the last few specimens from the Lindley collection, just because they are so interesting, and so beautiful. Here's an Iris imbricata. This is from the Caucasus Mountains. Here again, you've got some hand-coloured artwork. You've also got a little extra illustration. This is a Lindley illustration. He's done a little bit of dissection of the central part of the flower. And the label from the garden of the Horticultural Society, what became the RHS. So this was flowered in the garden in 1845, and this is the type specimen for this species. And here is a lovely Nepenthes specimen. This is Nepenthes albo marginata. This was collected by Thomas Lobb in Penang. Here you can see the picture of the plant. So this is one of the carnivorous plants. So this is the leaf, and the leaf extends into the structure, which has digestive juices inside, and insects and all sorts fall in, and the plant digests them. You can see the mottlin on the outside of the picture, which is quite distinctive of the species. It's a very beautiful one. So this is the type specimen, the type material for this species. And again, we have another previously undesignated type, which was found in the collection. This one is not in such nice condition. This has become very soot damaged for various reasons. And the Lindley collection contains quite a lot of material which is in this bad condition. This needs proper conservation work to preserve it. But here you can see these enormous pictures, and this is part of the type material for Nepenthes veitchii. We've got the original label from Thomas Lobb with the location and the altitude that was collected in the blue label. And then we've got a label added saying it was collected by Thomas Lobb in Borneo, and it was sent back to Veitch and Sons, horticulturalists. And Veitch sent material to Lindley to get the new species described, essentially. This is Lindley's handwriting, Borneo, Lobb, T. Lobb, and this is the specimen. And here you can see the gold peristome structure around the edge of the picture. I think this is a really beautiful specimen, but it needs some conservation work to preserve it. So that's a little overview of some of the historic specimens in particular in our collection. There's a lot more I could show you. I could show you a lot more from the 20th century. I've really concentrated on more our historical collections to start with, and some of the priority specimens which we will be making available for the public and for researchers online soon.
Thank you, Lauren, for the wonderful tour of the herbarium. And now I will open the room for you guys to ask questions to our lovely curator, Lauren Gardner. We can't hear you, Lauren, unfortunately.
Gremlins Day. Okay, can you hear me now?
Yes, we can.
I'm so sorry. As you will have all seen, we had some real gremlins in the technical side of things today, and we're so sorry that we started so late. And yeah, we're having some really bizarre translations in the automatic captions as well. So thank you for bearing with us. I am really amazed with how many great questions have been coming in. I don't know how we're going to get through quite so many of them, but we will do our best. Hopefully Sabrina can help collate some of them yes. And group them maybe a little bit, because there's quite a few common themes. So I'll hand back to Sabrina to maybe collate some questions and start asking me.
So you have said it right. We have really good questions here. So I'm going to start. And one of the questions that we had, Lauren, is, does the collection hold many fungi, and what is the proportion compared to the plants?
Oh, great. So that's a really difficult one straight away. So I hope that people might have got an idea from the tour how how much is not known about this collection? It's a big collection. It's 1.1 million specimens. That's the estimate. It's a pretty good estimate. I've surveyed the collection a few times to try to ground truth that number, and it's been cited in various places. I can't tell you for sure how many fungi we've got. I haven't really looked in depth. What I don't think I mentioned in the tour was really that it is just me. I am the only member of staff here looking after this collection, and we don't have a database. We don't have a catalogue of the collection yet. So this is something that we are going to be working on over the next few years, in particular, trying to get a handle on exactly what we have. We do have fungi. We probably have maybe several tens of thousands of fungi specimens, if I had to put a rough idea on it. What we do have is a lot of type specimens in that fungi collection. So it's an important, rich collection. So it's a priority to make accessible, like so much of the collection.
Fantastic. Now, one of the quiz questions that I have, and I think is really good, is it ever technically possible to preserve a specimens in a way that the colors are retained? Because some specimens have amazing colors.
Yeah, yes. So we do have some specimens which have some really, really beautiful colors preserved on them. We have specimens which are 200, 250 years old, which still have blue flowers, pink flowers, quite a range of different colors, actually, sometimes quite surprising. Some pigments do preserve better than others, some will degrade more quickly. It often is more related to how the specimen was preserved in the first place and how it's been stored since. So if you make a specimen carefully and well in the first place, if you dry that specimen reasonably quickly, but without cooking it, without scorching it, without letting the colours and the pigments degrade, then you keep the specimen dry and you keep the pests away, you can keep that specimen pretty stable for a long time, hundreds of years potentially. So sometimes it will be a combination of chance and conditions of collection and preservation since, but also the chemical composition of what those pigments are. It's a mixture of factors.
Fantastic. Now, quick question here. I'm gonna put them together. If a specimen is in poor condition, is there a program to source new specimens if possible? I mean, if at all possible, or do researchers just use those as reference materials?
So a lot of collections, a lot of herbaria have very active research programs where they are collecting material, they're working with researchers who are depositing specimens in their collections. Also, herbaria around the world and natural history collections more generally, they exchange material. With herbaria, with herbarium specimens, we tend to collect duplicates of specimens. So each time you collect a specimen, you don't just make one single specimen from that one plant, you often make multiple duplicates of the same plant from the same place at the same time. And then you distribute those duplicates to different places. So that you've got a physical backup in different collections. You know, in the days before we could be imaging specimens and databasing them and sharing those records, literally having a physical backup in another country helped prevent losing all the material in case of a disaster. But also it means that researchers can work on that physical material in country much more easily than having to travel or send the material backwards and forwards. So going back to the question, which was about adding to the collection and replacing material that maybe is degraded. Here in Cambridge, we don't really have an active programme of accessioning new material, collecting new material. We do accept some duplicates from other collections. At the moment, because it is just me, because we're sort of bringing this collection kind of more into a modern research collection, again, more active, hopefully we will be looking at doing that in the future, but it's adding to the collection is a huge part of our work at the moment, but it is important for many collections.
Lauren, are there any women collectors in our collection at the Herbarium?
Brilliant question. I wish I could tell you how many and who. Yes, we will, but I can't tell you right now. Again, because we don't have a database, because we don't have a catalogue, it's really difficult to tell. So the way the herbarium is laid out, the herbarium specimens are laid out systematically. So if you are looking for a particular plant, you go to the part of the herbarium where that plant is most likely to be. So if it's a British specimen, if it's a plant from England, you would go to the British and Irish collection, and you would look under the plant family, plant genus, geographic division within Great Britain and Ireland, and you would look for your specimens laid out like that. But that doesn't let you search by collector. It doesn't let you find out who your collectors are. It doesn't let you find out lots and lots of things. It's a sort of single access way into a collection. Once we can start databasing our specimens properly, we can start searching and filtering and pulling out all those names. There's a lot of work going on in lots of collections at the moment, really exciting stuff, where they are discovering particularly women, people in underrepresented communities around the world, native people, indigenous people who collected specimens. At the moment, unfortunately, when I list the collectors that we know we have, they are a particular demographic of individuals. There are a lot of very famous historical figures. A lot of them are called John, quite a few of them are called Charles. A student pointed that out, that literally it was Charles, John. Yeah, but we've got lots of stories to tell within this collection, and yes, we will definitely have those kinds of specimens by different people.
Fantastic. Now, there was here one about the illustrations. So sometimes there's illustrations on a given specimen. And the question is, are they drawings or are they basically illustrations still added to specimens nowadays? And there's another question that regards whether or not you trace on top of it, or is it just basically freehand illustrations?
Oh, it's a mixture of lots of different things. Most botanical art is drawn just from the specimen or from a living plant. Not traced or anything like that. And although there is some artwork in some collections, nature prints and things, which will be based actually more directly on the actual material itself, so a bit more like a carbon copy in a way. But most of the ones we've got will have been painted or drawn separately, looking at the original material, particularly in that Lindley collection, where John Lindley had artists, botanical artists, specialising in those kinds of illustrations, and those illustrations were often used to accompany the publication of the new species in the scientific literature.
I have a question here that is very relevant. It says, is anyone ever allowed to remove material for extraction of DNA, or is it not possible to get DNA from these specimens?
So back when I did my PhD 15 or so years ago, and I was doing phylogenetics, so getting DNA out of specimens, herbarium specimens were really, really difficult, particularly for the group that I studied, which is orchids. Really difficult to get DNA material out of orchid specimens for various reasons. And at that point, we were using just Sanger sequencing, where you needed long reads of DNA, you needed unbroken pieces of DNA to get good sequence data out of them. As specimens age, and particularly if they haven't been dried very well or they haven't been stored particularly well, the DNA will break down. So all those fragments will get smaller and smaller and smaller. So it used to be very, very difficult to get good DNA out of older specimens in particular, and specimens which maybe hadn't been collected in absolutely optimal conditions for this kind of work. So back when I did my PhD, getting DNA out of specimens was a nightmare. I did almost all of my work was from fresh living material collected in the field or in botanic gardens. Now, increasingly with the new techniques, actually the first step of those techniques for DNA work is to chop the DNA up into tiny pieces, which in a herbarium specimen that's quite old has already been done for you. It's not easy, but it increasingly can be done. Now, whether or not we would do it from all specimens is more of an ethical consideration. If we have one single specimen of a plant that has no other representatives in our collection or in any collection, and it's a small specimen, and taking a piece out of it for any kind of DNA work is going to retrieve, obviously is destructive. You're not going to be able to replace that bit that you've taken off. Then it is quite unlikely that that would be given permission. But most collections consider this on a case-by-case basis. If there's a good, justifiable reason, reasonable chance of getting good or some results at least, then yes, these things do get considered. And we have had examples where people have taken DNA from some of our specimens.
I have a question here that says, does the herbarium receive any specimens or donations from the general public, either antique or even fresh specimens?
Okay, so as I said, we are not actively adding to this collection at the moment, but we hope in the future we will have more of a strategic accessioning policy for adding to our collection. We do have modern day material. So in the tour, I focused on our more historic collections. We have extremely rich 20th century collections and the first 2 decades of the 21st century as well, because Cambridge is the centre of a lot of botanical activity. We have many, many botanists based here. It's an incredibly well surveyed part of the country, incredibly densely surveyed. And a lot of those researchers over these have left their collections to us in bequests, or they've deposited their specimens over their lifetimes. So the Oliver Rackham collection, for instance, is that kind of a collection. It's relatively recent. It goes back from 1950s through to well, shortly before Oliver Rackham passed away in 2015. I think, get my dates right. So we do have modern material added to the collection, but it's not in a particularly strategic way at the moment. Did I answer the question, or did I go off on a tangent too much there? Sorry.
I think you did. There's a question here about basically specimen. How do you deal with it? So one, what type of paper do you use for pressing? Does it have to be replaced regularly? Do you have to wear gloves to handle the materials, or do you have to do anything for freezing in regards with the water in the specimens?
Okay, so when you make herbarium specimens, and this is a whole talk, I could talk about how do you make specimens. All these questions are whole talks in their own right. Actually, I could talk forever. I am conscious of the time. We are at 2:05, so we're sorry if we are having to lose some people, but I think we could probably carry on for another 5 minutes, if that's okay. So making specimens, the very, very simplest level, most herbarium specimens are simply pressed in newspapers and in a giant flower press. So a wooden frame, a big flower press essentially, where you build up a sandwich of newspaper, plant specimen, newspaper, and then either side you've got corrugated card or corrugated metal. And you've got like a big sandwich of specimens builds up, alternating those layers. You squish them between your press sides, you bind them together with straps, and then ideally you have warm, dry air passing through the press to carry the moisture away. In the field, you have to come up with some Heath Robinson kind of inventions to make sure that you can get that moisture out and stop the specimens rotting, particularly if you're somewhere that's humid. So a lot of my work was in tropics, and it's quite difficult sometimes to dry your specimens well. But you generally change the newspaper frequently, pretty much every day for many specimens. You change that newspaper, so you're taking the moisture out of the specimens, you're pressing them, drying them, pressing them, drying them, and hopefully getting that moisture out before they start to rot. So the raw material usually is newspaper. Newspaper isn't perhaps ideal, it is acidic, but frankly, it's what you can get hold of pretty much anywhere in the world, cheaply and en masse, and it's the right dimensions for making specimens. So that's kind of how you make your specimens. Then if you've dried them properly, then you won't have moisture in the specimen and you won't have fungal growths on them. You won't start going moldy in the collection, and you want to make sure that that doesn't happen. Regarding handling with gloves, we don't generally handle herbarium specimens with gloves, and that's quite different for some areas like the art world and some other heritage areas, museums, where people do wear gloves more often. gloves tend to make you less tactile. It's more difficult to handle specimens with gloves on, and you're more likely to, most people are more likely to damage the specimen accidentally because you've got less sensation in your fingers and you can't be as delicate. If you wash your hands frequently, if you don't touch your face, if you don't eat your sandwiches in the herbarium, which you shouldn't be doing anyway, because you're very likely to bring pests in and you get greasy fingerprints on specimens, then you can make sure that you're not ingesting any toxins on the plant or any pesticides which have been used in the past on the specimens. So careful handling, washing your hands frequently, very congruent with COVID times. You know, herbarium folk, natural history collections people wash their hands frequently anyway, because you don't want to be ingesting anything nasty, but you also don't want to be transferring anything onto the specimens either. But generally, we don't wear gloves. No.
And I have a very good question. It says here, if you were making a specimen for the herbarium of a large plant like Musa or Strelitzia, you need a huge sheet of paper or you just bend the leaves?
Really, really good question. So I used to collect palms as part of my research, and some of them are huge, great big trees. Again, this is a whole other talk. I could tell you lots about this, but with those kinds of specimens, you ideally take lots of photographs. You make really detailed notes, which will be incorporated onto the specimen label, ideally, saying how tall the plant was, how big the leaves were, how many leaves there were, that kind of thing. But then you make a representative specimen that can fit in the herbarium. If you make one massive leaf into one massive specimen, you're not going to make friends with your local curator because they've got to look after that, and that's really hard. But even something like a huge palm leaf can be dissected down to sort of the core components with a big long pinnate leaf, a leaf sort of shape of a feather. You can chop off one side of it for starters because it's kind of mirror image on each side. You can take a section from the tip, a section from the base, and a section from the middle, and basically assume that the bits in between are sort of in between those things. So you've got 3 separate pieces for that one leaf, and then you can start folding the leaflets so that they would fit onto a sheet of paper or into a box. A lot of collections curate into boxes for that kind of specimen.
Fantastic. Now, I am aware of time, even though we started a little bit late, and we do truly apologize, all of you. We just had a bit of technical issues that we had to deal with. But I just would like to say thank you to all of you for all the great questions that you guys put in for us. And also, I would like to say bye-bye. And I guess Lauren will say bye-bye to us all from this amazing tour that we had today of the herbarium.
Absolutely. Thank you so much, everybody who's joined us. Thank you for bearing with us, and we are really sorry about the technical difficulties.
Pleasure.
This is the first time I've been involved in lots of webinars and meetings on Zoom, but this is actually the first time we've hosted one ourselves. And as you can tell, it was a bit more complicated than we thought. That the real trick was trying to do the video and have it not jerking, not having the image completely jerking, but we couldn't get them synced up very well. So we're ever so sorry about that. However, we have lots of other events going on. We've got another one starting in about 15 minutes. That one doesn't have a video, so we won't hopefully have any of the same problems we had. But thank you. But do check on the Herbarium website, follow us on Twitter. There's lots of ways that hopefully you can find out more about the Herbarium in the future. And as we make it much more active, we will be able to share a lot more with you. So please do watch this space. At the moment, our website is quite basic. We have a new one in preparation, and our herbarium specimens, getting them digitized, getting them online is going to be a really important piece of work over the next few years in particular. So thank you. We hope to show you lots more in the future. Okay, thank you very much.
Thank you, everybody.
Bless him.